Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we would like to take a moment to thank our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Please mark your calendars for our next Dr. GPCR symposium held on Friday, September 22nd on the topic of GPCRs as therapeutic targets. The symposium is free, but you must be a Dr. GPCR Ecosystem member, which is also free. Please see the menu button for the symposia page at the top of our welcome page to check out the program and register as a presenter. You don't have to register as an attendee. You just have to be part of the Dr. GPCR Ecosystem to get access to the links to join us. We also have set aside 2 hours for networking and poster presentation on Kumo Space that day. The posters don't have to be fully formatted. Just come with a few slides and have a chat with your community members about your favorite GPCR or other signaling proteins that you're working on. If you'd like to present during your work during the networking time on September 22nd, please complete the submission form available on our website. There is no limit on the number of posters we can accommodate. To present, you'll only need to provide us with the title of your presentation, a 1-minute video abstract, and come with a few slides to help you discuss your work with your colleagues. Everyone is welcome. If you have any questions or comments, or if you'd like to present your work but don't know how to join us, please feel free to email us at hello@drgpcr.com. What are you doing between November 2nd and 4th? Join me and the Dr. GPCR team at the 22nd edition of the Great Lakes GPCR Retreat at Chateau Montebello in Quebec. We at Dr. GPCR are proud to be supporting this iconic meeting. I've had a blast help organizing this meeting this year with my colleagues from the University of Ottawa, the University of Montreal, and the University of Sherbrooke. Visit gpcrretreat.org for more details on the program. The early bird registration and poster submission deadline is September 8th, and the late registration deadline is September 21st. Hurry up and join us between November 2nd and 4th. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and this morning I'm very happy to have with me Dr. Evi Kostenis. Evi and I met recently at the Gordon Conference in person for the first time, although we've interacted previously, so I'm very, very excited to have you on. Good morning, Evi. Or good afternoon.
Good noon, Yamina. It's very nice to see you and meet you here again, and I'm looking forward to our conversation.
Thank you so much.
Thanks for choosing me, by the way.
Thank you. And thank you for accommodating. I know both of us have been busy, so I think we've been postponing this call for months now, but we're finally here doing it. Very exciting. All right, let's start at the beginning. Would you please introduce yourself and tell us what is it that you do?
Okay, my name is Evi Kostenis. I'm a pharmacist by training, and couldn't imagine I would ever become interested in GPCRs. So pharmacy study, PhD in pharmacology, postdoc, everything GPCR related. And I had many, many different steps in my career. Finally, I was also at industry, big pharma, small pharma for many, many years, and finally ended up at the university. But all my life, I've been working with GPCR. So now I'm, of course, a professor at university teaching, and I have a lab and an institute here where our group is taking very much care of understanding how GPCRs function.
Thank you. That's very interesting. You mentioned, I didn't know that you were in industry as well, and large pharma, and then getting back to university. I'm very interested in hearing a little bit more about your story, because typically what people think about is you start in academia, so you do bachelor's, master's, PhD, postdoc, and then if you go to industry, coming back to academia is a very difficult step. And
That's
it turns
true.
out.
Yeah.
And I'm really interested in hearing a little bit more about your career.
Okay. Well, yeah, my career is extremely weird and I think more rare. So studying pharmacy means that you have in mind going to a public pharmacy to work there or in a hospital. And I did all these studies and I ended up in a pharmacy, both in a hospital and in a public pharmacy, and I was bored to death. So I resisted there for 2 years, but in parallel, I was working for newspapers when new medicines were introduced into the market, and there were press releases, press conferences. So I did something to not be that bored. After 2 years, I couldn't stand it anymore. I insulted the wife of the owner of the pharmacy, so I had to leave it. So that means I had to do something else. I would have left anyway. And then I remembered I really liked pharmacology. And many GPCRs are part of pharmacology because so many medicines act through GPCR. So I was trying to find a place to do my PhD. After that 2 years of work in a pharmacy, I found it at the University of Bonn, and I really enjoyed it. And then, I mean, and there, yeah, it was allosteric modulation of G protein coupled receptors. And then I wanted to learn more molecular stuff. And I did a postdoc at the National Institutes of Health in Bethesda, USA, 3.5 years in Jürgen West group, which was same receptor family, but completely new aspects. And then I wanted to go back to university, but I couldn't because I followed my husband to Frankfurt back in Germany, but their university was so terrible that I chose to go to industry. And I ended up in the cardiovascular disease group, initially as a postdoc, and a few months later as a lab head, being allowed to work with G protein coupled receptors in the cardiovascular area. And there after 2 years, they wanted to push me into a management position, which I didn't want. And they told me, oh, you will get a car and more money. I said, I don't want a car. I want to do science, and I won't be able to do science. And then they were forcing me to take on that management position, but I didn't want. And so I chose to leave the company and go to a small biotech company in Copenhagen, Denmark, called 70M Pharma. Guess what they are doing? 70M receptors, right? And but that was so 4 years big pharma, then 3 years small pharma. And all this time, I was trying to keep connections to people in academia, trying to publish to stay alive so that eventually I could go back, because going back was really, really difficult. And then there was, I think, a lot of lucky coincidences that made me apply in Bonn University and even get the job there, because I was not number one of the list, I was number two, but number one had another offer. So number two is number one, and I took it because I thought it's maybe the only chance in my life to escape from pharma and try to see what you can do in academia. And there I am since 2006. Ever since, I haven't changed, because I think of all the things I have seen, academia is still the most enjoyable. And yeah, so this is how it happened. And I know that often you say, oh, once you leave academic research, you will never go back, which is true if you don't publish and you have no connections. But if you manage to have something on your CV, some kind of publications, you may manage to go back.
That is fantastic. And the reason why I was pushing you to tell us a little bit more about the stories, because most of our audience is in the younger generation, the up and coming early career scientists who are currently either finishing up a postdoc or just started, their job at a university or even their job at a company. And I think that's very important. You made a couple of very interesting points. One is the fact that you need to continue publishing if you can. I think biotech and pharma have changed in the past decade, and they're pushing to publish because it looks good for the companies as well. The other thing is keep your contacts, and that's all about networking, staying in the pool where you know people who can give you advice, who can help direct you, who can put in a good word for you. I think that's very important, and independently of your level, you need to be doing that, at least in my opinion.
Yeah, I agree. I mean, I never had people helping me to find contacts, but as you move along in your career, you see how important it is to have friends and contacts, and they will help you at some point to move on.
Yeah.
It's extremely important. And now when I have my own students, I tell them, go to meetings, meet new people, meet your future collaborators. And if you see them, you will also behave better and more fair when you know the people you are dealing with. So create your network and be fair to have as many connections as possible that you will need in science to survive.
Absolutely. And I think it goes both ways as well. So one is you never know who will be able to help you, but also you have to think about how can you help someone else.
Yeah.
I think that's very important. It goes both ways. It's not a unidirectional type of relationship, you get to help people. If you can put in a good word, if you can give them some advice about their project or about the direction, or just, let's say you read a paper and you see a poster presentation and you ask the person, did you read this paper? This is a really good paper that could be relevant to your science. That can be helpful. And
Yeah.
that's also a level of, helping out and networking, because the
Yeah.
person will remember you. Oh,
Yeah.
that's the, that's
Yeah.
the one who gave you that paper.
Yeah, I absolutely agree. But this is also what I teach the students, to be as helpful as possible, because if you are helpful and you discover others are just parasiting on you and never giving anything back, you will not help them anymore. But I would say, in the first place, you would always try to be as helpful as you can, hoping that the other person appreciates this and functions the same way,
Yeah.
because you don't want to be abused and you don't want to be the idiot. But I would always help and support people, and then
Yeah.
hoping of finding a network of people that you can rely on at that, and that will help you when you need help.
Yeah.
And if people approach us, can you support us? That yes, we can. And so it's like you try to construct some kind of win-win situations. So not,
Yes.
okay, I'm going to abuse you as much as I can, because now, it's, and
Yeah.
this is the opposite of what I think you should do. You always have to search for interactions that keep on existing because of the win-win for everybody. That's,
Absolutely. No,
yeah, yeah,
absolutely. I think that's what I meant by going both ways. And obviously it has to be balanced out.
yes.
And yes, obviously always asking or always giving and not getting anything back. That's not a, I think I always tell this to people. It's, building a relationship, no matter what type of relationship, it's like dating. You have to like each other, and everybody has to weigh in and put in, something into that relationship to keep it going, whether you're looking for a
True.
job or for your next mentor.
Yeah, that's true. Yeah.
So you mentioned starting out in pharmacy. You're not the first guest on the podcast who has a degree in pharmacy, who worked in a pharmacy and did not like it. You mentioned that you always liked molecular pharmacology. Do you remember the first time you heard about GPCRs?
Think during my thesis, when, oh no, wrong, in my studies, of course. I mean, if what you read since 50 years or almost, or that many years, that GPCRs are or 30%, well, it was 50% of drugs on the market, actual GPCRs. I think it would be terrible if I said now that during my PhD time I heard for the first time about GPCRs. So that means I must have heard about it before, but I just don't remember this
Mhm.
actively. Yeah.
No, that's okay. It happens. I had the reason I was asking is because I do remember the first time, and we had sometimes guests who said I knew nothing about GPCRs. I just wanted to work in this lab because I thought it was a very interesting topic, or I liked the city, or I liked the PI, and then they ended up working on GPCRs. We all have a different story.
In my case, it was different. When I was working in a pharmacy, I was going to the yearly meetings where different professors were giving talks. And the professor I was fascinated by, because his talk was so good, was the professor who then started his career at the university at Bonn, just when I applied. So I was his first PhD student. And I didn't think of the research. I was fascinated by the teaching, because his class, and it was about liver cirrhosis, which probably doesn't have much to do with GPCRs, and is a topic that sounds perhaps boring, but he made this so fascinating that
Yeah.
I had to approach him after the talk. I said, okay, can I do my thesis in your lab? Not, I just wanted to leave pharmacy and I wanted to do something else. This was a pharmacologist. So I approached him, and
Yeah.
then his subject was allosteric modulation of muscarinic acetylcholine receptors, which has nothing to do with, no matter what it is. I think you just have to have some kind of interest in science, and then many things can be offered to you. And if you are like curious, you may like all of them.
No,
Yeah.
absolutely. I agree. I agree. And it's such an interesting point that you made about, watching somebody teach and really liking the way they teach. I think, and in my case, for a very long time, when I used to go to conferences or to take courses, I always felt like I didn't sometimes I didn't understand what the teaching or the talk was about. And then I realized it's not me, it's the way it is presented. And
Exactly.
some have this beautiful way of engaging the crowd and presenting. And no matter what the topic is, you're just fascinated. You can't take your eyes off of those slides because the person is so engaging.
Exactly, and that was the case. When he accepted me as a PhD student, I was sitting in his lectures for the students, although I didn't have to, because I was just fascinated by the way he was presenting and explaining pharmacology. And there you sit and say, okay, this is my example. I mean,
Yeah.
I can never reach this example, but you are sitting there fascinated by the way he transmits his information. And his lectures were in the morning at 8 o'clock. You would expect no student to be awake at that time. The classes, the lecture hall was full because of
Wow.
this guy. So, and when I studied pharmacy myself, I mean, nobody was sitting there. There were three people, and eventually no one anymore. The professor was alone because the classes were just dreadful. And
Mhm.
yeah, so it's about how people present things. And now I'm telling my students, oh, go to a good lab, think about what you like and think about whether you like the research, you're excited by it, and then go and choose because going to the right lab may be pivotal for your career.
Yeah.
At my time, we were thinking different. We were also not so focused on good labs. And I mean, I was absolutely not focused on a good lab. I was just fascinated by someone who was teaching so well that, you want to learn how to do that.
Yeah. Yeah. And I think that also shows the passion that the person has for the topic and for teaching and for helping, which most of the time translates into how that person is in the lab as well. And I think to be able to give an amazing talk, a very good talk where you're engaging takes so much work because you have to practice, practice, practice to get to the point every time and keep the audience engaged. So that's also shows talent but passion as well for that. So tell us a little bit more about your PhD thesis. So you mentioned allosteric modulators. When you started your PhD, did you pick the project you wanted to work on, or did you develop it with your PI?
You're asking me about something that is 31 years ago. That means you know now how old I am.
I'm
I think
taking you down memory lane.
okay, but yes, I had a choice because I came and there were three projects to choose from. And one project was, now let me remember, structure-activity relationships of allosteric modulators acting at muscarinic receptors. There, a chemist professor was synthesizing the molecules. The second thing was to exclusively analyze alcuronium-type allosteric modulators on isolated atria. And the third thing was to study allosteric modulation depending on the ion composition in the buffer. And
Wow.
exactly, wow. And that's okay. So, not knowing anything, I mean, I had no shitty idea about what all this was, but I thought, well, structure activity relationships of allosteric modulators acting at muscarinic receptors is perhaps the best option. So I picked this. And it was because after 3 months, I already had my first poster on a meeting where I could show results, whereas the others have been struggling quite a bit until they could first show their results. So I
Yeah.
was lucky. I didn't really know what I was choosing.
Mhm.
I thought I chose the worst, the best of the worst options, but in the end, it was fine. Yeah.
That's great. And then at the end of your PhD, you mentioned moving to NIH and working in Jürgen West's lab. How did you make that decision? It seems like throughout the conversation that you were very keen on pharmacology and working in a lab and working in science and in academia. What made you decide to move abroad?
Um, I loved what I was doing, but what I was doing was extremely limited. So the methods we had access to and the money in the lab was not much. And the methods we had access to was just binding assays. So we did a lot of binding membranes from hearts that of cows and pigs, and so no cells. It was all animal hearts that we needed and to convert into membranes and do binding studies, associations, dissociations, equilibrium binding studies. So pharmacology up and down, the classical pharmacology. And we had to kill some guinea pigs and open the thorax, or however you call it,
Yes.
whatever this.
Sternum. Yeah.
And then to excise the heart, prepare the atria, mount them in an organ bath, and then keep them, watch them contracting in the presence or absence of the modulators that you are acting. So we had these two methods for 3 years. I know I have n equal to 66, meaning I killed 66 guinea pigs. I still remember that
Oh
because
my God.
it was hard to do this, as you had to kill them all the time, and then even I had to kill them for the other students who didn't want and or who couldn't do this. So, but I don't remember the n number of my binding studies from animals which I haven't killed because we got the hearts from the slaughterhouse. But the method spectrum was extremely limited, and I loved muscarinic receptors, and I was reading, but the methods were it was so few that I thought, okay, I would like to stay in this receptor family, but learn new methods. And at that time, molecular biology came up, chimeric receptor signaling, and Jürgen West was the one that was really a pioneer in setting all this up for muscarinic receptors at the National Institutes of Health. So I was applying there, and he was interested in me, so I could go there and continue as a postdoc, but sticking with the receptor family, just expanding the methods, which is actually something I also tell people if they would like to develop, like broaden your method repertoire, learn more, to ask new questions and have like a big spectrum. So it was just to increase the spectrum. And my supervisor was really worried because I said, I want to go to this lab and I'm not going anywhere else. And then he said, Mrs. Kostenis, you don't have to be so obsessed with this one lab. There are more labs in the world you may go to. I said, no, I have to go to this lab. I have to go to Jürgen West. So I go there or I go nowhere. So he was worried that I was obsessed going there. And I applied for a fellowship. I didn't get it. I applied again. I got it and I could go there. And then I was there for 2 years. And then finally, Jürgen offered me a position to stay there a bit longer. As I have been less useless as he probably thought. Anyway, this is how it happened then. Yeah.
And how was the change from, moving from Europe to the US? Did you find it? I'm trying to, so the place I'm trying to get to is, again, knowing the audience who's going to be listening to this. I like to capture how people adapt to change. We all agree that it's important to go abroad or to change your environment as much as possible when you go for a postdoc, because it's important for you as a person to grow, but also as a scientist to show that, plopping you out of your regular environment as a PhD, going into a new one, you can still succeed and you can continue collaborating. That's why I ask these questions. I'm really curious to see, what did you think about moving to the US from a cultural perspective, from adapting to a new environment?
I didn't think so much about culture because culture was very different. I was more thinking about the science because at the time I went, it was like, by definition, you had to go to the US to be able to find a job later back in your country. It was just a must. I think now research in Europe has gotten much, much better. So you don't have to go to the US. There are other good labs in this world. It doesn't have to be the States. But at the time I was there, it just looked good in your CV. And coincidentally, it was the lab where I really wanted to go. So yes, you go. You give up a lot of things in Europe, all your friends, your family, because you are gone. But I think it also helps you and strengthens or shapes your personality because you are in a completely different place. Everybody's gone, everything's new, and you have to survive there. And suddenly you have colleagues in the lab from all kind of different nations with nice or weird behavior. Well, it makes you stronger, it helps you survive. So the lab environment was very different, much more harsh and much more elbows. I'm a postdoc and I want to be better than you. Before we were like more working together, and suddenly we have competition and even sabotage. So, so it's just the world. And but it was a really important move because it had to be a part of your career. And the culture is something, on the side, which is very different. I enjoyed being there, but I couldn't imagine to be in the US forever because I think that I'm too German or too European to be more at home in Europe
Yeah.
because of cultural reasons.
Yeah.
Which doesn't mean that I found the time fantastic and I would like to go again to the national parks. And this
Yeah.
is, I mean, it's such a big country. Everything is so big. Maybe everything is too big for me, and I feel cozy at home where things are smaller. But no, the culture is not really mine. But if you're from Europe, you don't meet so many Americans. You meet people from everywhere and not
Yeah.
Americans, which I think was great for opening your horizon.
Yeah.
But yeah, so the contact to the real Americans, we didn't have it. But you see, of course, how people live there. But I wouldn't go to the States because I want to live there. I
Mhm.
went there because working quality was excellent and I needed it.
Yeah.
Yeah.
And it's interesting you mentioning, how people are different and everything is big. That is true. Everything is big here in the US. And including the country. And what I do really like is that depending on which area in the US you go to, it's like you go to a different country because people are different even within the US. And
Oh, yes, absolutely. I mean, if you're at the West or the East Coast or the center, it's so different.
yeah, yeah,
It's so
yeah.
different. Yeah.
And it's important, you mentioning that you preferred going back to Europe because that's what resonated with you. And the reason I'm bringing this up is because it's important to do things that resonate with you. And this is more for the audience around figuring out what is it that you want, what do you like, what do you need for your career, and then remaining true to yourself, basically.
Yeah, yeah. I mean, in a way, you make a sacrifice because you give up all your friends and
Hmm.
everything, and there you are in the US.
Yeah,
Maybe now you would be contacting your friends through Zoom, but I'm not such a big fan of these virtual relationships. I prefer real people to
yeah,
talk to,
yeah,
we
yeah,
couldn't do this digital contacting and socializing at
yeah,
that time. So you were really alone. And I mean, when I was there for 8 months, having worked every Saturday and every Sunday, maybe 80 to 100 hours a week, I got so homesick and I said, okay, I have to go home right now. I just have to leave this country. And then I went home and after 2 weeks I recovered. Of course, I went back, right? But it was, yeah. So it's, so you're struggling, and it's not that. I mean, yeah, you go there and everything's fine, and no, you're suffering, you're struggling, everything is new. And then,
yeah,
yeah, I got homesick once, and I'm just telling this so open because maybe it happens to others, and it's not to feel bad about it. If you feel homesick, go home. Thanks God I had a boss who allowed me, because there were other bosses that were telling the Europeans you can maximally be out of the lab for 1 week. You have to come back. I mean, if you go to Europe, you travel one day back and forth, it makes no sense. But
yeah.
my boss was different and he understood it and he let me go away. It was fine. And I never had a second attack of being homesick again. It
Yeah.
was then fine. Yeah.
Yeah, and I think it's important as well to goes back to picking the right lab where you can have this really good relationship with your boss who allows you to do these things. And you're right, traveling from the US to Europe for when you have just, 5 to 7 days, it's just not worth it because you're most of the time flying and you are barely home for a little bit and then you have to come back. So under 2 weeks, absolutely not worth it
Yeah.
in that sense. All right, we talked about this a little bit before hitting record about your favorite GPCR. I'm still going to ask the question because I'm curious. You mentioned that you don't have a favorite one, and maybe I'm giving out the answer. But um, do you which if you had to pick a family or a receptor or anything related to the field as a pharmacologist yourself, what would that protein be?
A G protein.
Perfect.
I mean, but I mean, I saw the questions and I read the question, what is your favorite GPCR? And the honest answer is, I have no favorite GPCR. I like all of them. And what fascinates me about them is that they activate G proteins, so the way they signal. And if I try to think about it, I wouldn't be able to identify the GPCR. For me, it's just a means to turn on something that happens in the cell, because what I really like is what happens after activation. So any receptor could be a means, maybe not adhesion receptors, which are, well, nobody
Well,
really knows how they function, and they wouldn't be the prime example of turning on a signal because we still
yeah,
have to understand how they work. So I find that family fascinating, but it doesn't help me to switch on signaling, because what
yeah.
I really like is the signal transduction downstream of them. That is what I like. And there, I think I'm a big fan of G proteins as they do the signal, and of course, I mean, of signaling mechanisms in general.
Yeah.
Yeah.
Any favorite G protein?
My favorite G protein,
Yes, if you have one.
or maybe I'm biased because we have an inhibitor that helped us study that G protein family a lot, and that is the molecule FR inhibiting GQ and 11 proteins. So if I had to choose the G protein family, I think I would choose GQ, but I like all of them. Also the ones that are more challenging, like G12, 13, where we know less
Yes.
about it. So I love all of them, and yeah, favorite is GQ.
DQ. Yeah, I think Silvio also had a G protein as a favorite, but he also told me that it's difficult because he said, how do you pick your favorite? It's like picking a favorite child, which is
Yeah,
not an easy task.
that's true. Although every parent has a favorite child, they just don't say it.
Maybe. I don't know. It's a tough question. I have two. So if I had to pick, it would be a very difficult choice. So you finished your postdoc at NIH, then you came back, worked in industry a little bit, and then you ended up working at the University of Bonn, was starting your lab. How was it to come back from industry to academia, and how did you build the scientific projects in your lab when you started out?
That was really difficult. And even when I talked to the dean before coming, he said, I don't see how you will ever manage. You have not been in academia all your life. You have been in industry for 7 years. You're not bringing any projects. You are bringing nothing. You have to start from scratch. You have to build your lab from scratch. You have no co-worker who can assist you in getting everything started because yeah,
Yeah,
so usually people grow with you somehow and then they
yeah,
help you to build the business. And he said, I have no idea how you will manage, but well,
yeah.
um, let's see. So I thought I will manage. I didn't think about all the difficulties, but I was facing them when I was here because I had no grant money. I had a miserable budget that could just pay the toilet paper and the electricity per year, but nothing else, and maybe one FedEx shipment. So it's like, so it's really tough. And I came into labs, I think, which have had a very, very low level equipment. Many things had to be thrown out. And then it took like almost 3 years until all these renovations or renewals had taken place, until we finally could work there. The first PhD students, I had to put them in the labs of other people to see what kind of project I could possibly invent to make them work in another person's lab. I think it was really, really, really difficult. But in the end, you manage, and it just takes longer until your first paper is there. And, you know, it's just as you have to start from scratch, truly. It just takes longer. And luckily, I had the first PhD student is also someone who started studying 10 years later, who was a chimney sweep before and a truck driver,
Wow.
and then studied. And he was supporting my research so well. And we got we got a lot into label-free analysis of signal transduction, and that was a big part of the initial aspects of my work. And there, this guy ended up. Of course, it took him 5 years to finish his thesis, but he had a paper in Nature Biotechnology, and I think 8, 9, 10 papers in the end. It took long until the first one was there, but then things began to move. But the period in the beginning, I think, was very, very tough. And
Wow. Still fascinating. Chimney sweeper and truck driver turned PhD, a very successful PhD with a lot of publication. See, this is why I like the podcast because you get to learn things that you cannot appreciate by just reading a paper. And it shows we're human.
yes, I mean, and that paper, you know, I mean, the first paper at my own my own last author paper at the University of Bonn was in JBC, and we were so happy. Or was it in Nature Biotech? I don't even remember. Or So the first or the second one was JPC and Nature Biotechnology, and it was just incredible. We couldn't believe it. And this truck driver, chimney sweep PhD student that I had, he was putting such an effort. He fell asleep here in the library because he was working so much for the revision, because we couldn't believe it that we had a paper in such a journal in revision. And we were
Yeah,
both working day and night,
of
and
course.
it was amazing the effort he did for it. And yeah, but in the end, everything was fine, and the effort was worth it. Yeah,
Absolutely. That's fascinating. When you get to put in so much work, you start from scratch and then it pays off. I think in research, the one thing that we don't know is when will it pay off? Is and that's
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 yeah,
but
yeah, absolutely.
you have to keep going at it. I always tell people that in my case it was always from dopamine rush to dopamine rush, where you have finally something that works, and then it's like walking through the desert for weeks, months, and then you get another dopamine rush, and at the end you have a paper, but it's very difficult. We have a very difficult field, and you have to be, as a scientist, very passionate, very patient, and being ready to come back to the board if something doesn't work and start
Yeah,
all over again.
yeah. It's to be passionate and to resist and to be patient. Absolutely. Yeah, I agree, but that's a feature you have to have as a scientist, because if you're not, well, first of all, passion is what you need, otherwise you can't do research.
Yeah.
And to have patience until you finally succeed, because success is nothing that comes when you submit your manuscript. There is so much luck and so much lottery and so much, I mean, it's out of your control. You'll see what happens. And often you don't receive comments as you would have hoped for, because maybe your enemies are there, or people don't want you to publish what you try to publish.
Yeah.
And then, yeah,
Yeah.
you have to deal with it until you finally have a good end.
Yeah, no, I agree. And I always when I think about it, I always try to tell myself that the only thing I can control is how I react to
Hmm.
events. I can't control what happened. I can't control what is going to happen. I can only control how I react to things, and how I keep myself and my decisions and my feelings in check. And come whatever may, we'll figure it out. But
Yeah,
it's sometimes very hard.
yeah, but you control how you react, and you also control what you do. I mean, what you
Yeah,
produce is something that is absolutely under your control. And
yeah,
what I keep telling myself and also the people is, I mean, do your job as good as you can. If it's good enough, somebody else will decide. But don't regret if you get a no, because you could have done better. I
yeah,
think give your best all the time. I think that's the minimum. If it's good
yeah.
enough, we don't know, right?
Exactly. And sometimes with, publications and reviewers, it's a hit or miss. You don't know who's reading your paper. I've always thought about, it would be really nice to be able to submit publication papers to journals anonymously, so
Yeah,
that whoever's reviewing doesn't know who the authors are.
yeah.
To
Well, that
see
makes
to
no
see,
sense.
I know, but to see
Yeah,
if how people would react to it, or if you would get a different type of review, if that
yeah.
would be the case. Tell me a little bit about that GQ inhibitor and how did you discover it? Um,
We didn't discover it. I think we had, um, we had, I was offered a label-free biosensor to analyze GPCR signaling. And to analyze GPCR signaling with a label-free sensor would require that we can switch off each individual pathway to know what we are actually looking at.
yeah,
And the only tool available at that time was YM25489, a GQ inhibitor that you just couldn't get. There was a Japanese company that I found it, and they wouldn't give it, they gave it to a few people. I have been begging and begging, I never got it. And
mhm,
then I found someone who gave it to me and who was then part of our planned paper. And the paper that we then published to show how to visualize GPCR signaling along all the four pathways was only possible because we could switch off GQ signaling.
yeah,
That otherwise we wouldn't have had a tool. And so it was published in Nature Biotechnology only because of this natural product. And this natural product was inaccessible to us at that time. But then a colleague here in the institute made a structure search and found a paper that nobody has seen, telling me, look, this something looks so similar to what you were using, because I was absolutely fond of this YM molecule, the GQ inhibitor, and the other structurally similar ligand was had a name MH, and then some kind of numbers that I don't even remember. So that in the end was FR. Our GQ inhibitor, and we are often associated with it, but we haven't identified it. It was identified 1988 in Japan. We just re-identified it and did something with it, because this one you could extract from leaves of a plant that, you could just buy dry leaves of a plant. So you isolate the molecule, and then we finally had it in our hands to use it. So it was that experimental data with a structurally closed analog made us realize how fantastic data you produce with this molecule. And then we were searching for something comparable. We had it. And this is when research began, because switching off GQ was giving us an immense amount of options of projects. And this is how it all started, no?
Wow. That's fascinating, because it's such a great little, it's a good tool that allows you then to use it. And it's important in the field in general to have the correct compounds and the correct tool compounds, actually, to be able to.
And I mean, for us, it was also so important because I was interested in, at that time, G protein independent signaling. And I was always surprised, how can people claim G protein independent signaling if the only tool we have is pertussis toxin? That nobody has ever switched off all G proteins. And then, it was just the next step. Take away 2 G proteins downstream of a receptor, and then you're still lacking GS and G1213 that we can't yet inhibit with anything decent, probably. So it was also, as I said, I'm interested in GPCR signaling, and I'm interested in G protein inhibitors because they can take away a transducer of a receptor. So that allows me to see if there is any kind of signaling in the absence of active G proteins. And I think that our papers on FR and all I think I have to say different. I think we really revived the G protein branch of the signal transduction field by bringing FR to the field and by focusing again on G proteins, because we were able to take them away. So you take them away, and then you see what else is left, and then you discover there is no signal left. So what is G protein independent signaling? And there you go, and things get interesting.
No, I think so too. And I remember trying to do to CRISPR out or to knock out selective G proteins in HEK cells, and then HEK cells actually, since you had other G proteins, then they would couple to the other receptor because the cells would adapt. So having a chemical tool that allows you to really inhibit the interaction is a really great tool and an additional set in the toolbox.
Absolutely. And I think, the pharmacological inhibitors like pertussis toxin and FR or YM, in combination with the tools developed initially by Asuka Inoue in Japan, all the knockout cell lines where you CRISPR away the arrestins or all G alpha proteins. These were incredible to also reinspect, like paradigms in the field, like G protein signaling, arrestin signaling, because if the G proteins are gone, you have to see arrestin signaling if it's a standalone pathway. And that was like a big discovery. Oh, there is no arrestin signaling. And what we thought was arrestin signaling is actually G protein dependent ERK activation. So the prime readout considered as arrestin signaling is G protein initiated. And these are like, wow, we only know because of the tools, but retrospectively, I think, in the absence of any tool, because we only had pertussis toxin. So it's amazing how a field develops in the absence of data to support the hypothesis. Anyway.
No, agreed. I know we're winding down a little bit. Having worked in industry on GPCRs, worked in academia on GPCRs, I always ask this question. I know the answer to it, 100% yes, but I'd like you, if you could elaborate on it. Do you think GPCRs are still good drug targets?
Yes, I think they are, and we have to change the way we consider how they function. I think we have been not really successful in the last 2 decades because we are trying to make drugs that have wanted effects and no side effects, with the idea
Yeah.
of separating signaling through one pathway, which is causing a good or a bad thing, and signaling through the second pathway, which is causing the opposite. I think this thinking contributes to having no drugs. All
Mhm.
this biased signaling big thing which is there. I think this is the reason for failure, and we have to get away from it to remembering that the initial GPCR drugs were not found by knowing the target and by dissecting the
Yeah.
pathway and by thinking about wanted and unwanted effects. So, these drugs were put in animals and you see a reaction that is
Yeah.
interesting. So the approach is completely different. And I think that GPCRs are still good targets because they are sitting in the membrane and they are good to reach. But I think the way we are trying to address them has to change.
Yeah.
And yeah, this wanted pathway, unwanted pathway, side effect, wanted effect separation, I think this is all thoughts that won't help us, that just explain why we have no medicines. And
I
if we
think,
forget this, we'll get drugs again.
yeah, well, possibly because I think they function. So I think GPCR's function is so complex, and when we study them, typically it's one GPCR, two, three pathways, and then that's that. But they are expressed in cells that express other GPCRs that are part of an organ, that are part of an organism. So it's very difficult to say pathway one versus pathway two. I think we need a way to, yes, you have to quote unquote simplify it, but at the same time, you have to keep in mind that whatever drug you're developing is going to end up in a human being, and humans are
Mm,
complex organisms. And if we could refine or take a more holistic view as to what does that GPCR in the context of an endogenous system where there's other signals to be taken into account, that might help us get better drugs faster. But I agree, I think it's very difficult to say yes or no. I just think about what a GPCR has to do in the context of a cell endogenously in an organ, in an organism, it's hard to say, oh, it's activating this G protein versus that G
mm,
protein versus
mm,
that. It's a
mm.
it's a holistic reaction to
Yeah.
the environment.
Absolutely. I think it's not the right way to think about pathway one here and pathway two somewhere else. I think my impression is that the GPCRs that control major organ functions, like like maybe blood pressure, heart rate, whatever, they have already been found. So
Yeah,
now, and they, angiotensin receptors, beta receptors, there are drugs there, blockbuster drugs, and
yeah,
so it's like the relevance they have is so, I mean, so obvious, and
yeah,
their utility as a drug target is so obvious that, and these obvious ones have been used. Now there are some others, the function of which is much more subtle, and
yeah,
maybe they play a role only under certain pathological conditions, or it's much more difficult to bring their relevance about, and
yeah,
equally difficult will it be to find a medicine and the type of people that may benefit from this medicine, because
yeah.
I think the big how you say um, probably you know what I mean. It's like a small screw that you turn slight,
Yeah, I know what you mean. I'm having trouble with words as well. You
anyway.
mean the oh God, I forgot the word as well, but I know what you mean.
Yeah. Okay. So it's like, yeah, I mean, to lower blood pressure, we know what to do. But maybe for a very specific, I don't know, I don't even have an example now, we wouldn't know which receptor perhaps plays a role. And then
Yeah,
the amount of people that could take this medicine would also be much, much lower
yeah,
than
yeah,
the huge group of people which suffer from high blood pressure, something like this.
yeah.
No.
I
And
think I think yeah, please.
yeah, no, no, sorry, you.
No, I was just gonna say that yes, I think there's a couple of things. We know a large number of GPCRs that are implicated in the most or the most not necessarily the most relevant in very relevant diseases, and
Yeah.
those are either are being targeted or we have tried to target them. I think we need more tools to uncover the less obvious GPCRs. We need tool compounds, we need systems to figure out. I think I'm fascinated by adhesion GPCRs, as you also mentioned them, but also by orphan GPCRs. I mean, some of these, we don't know what they do. We don't know where they're expressed. We don't have the tools to detect them. We don't have the tools to study them from a function perspective. And then the other thing that it's a topic that Madan Babu really is great at. He presented at the GRC as well. It's the variance, it's genetic variants in the human population. So you might have the same receptor, but you have a different mutant, or you have a different genetic variant. So the one drug might act differently on two people who might have the same disease, but the GPCR context changes. And I think that's also very important to take into consideration.
Yeah, yeah, it is, but I'm not sure if this explains why fewer. What is this? I don't know. I think I have to maybe unplug my phone now.
No worries, take your time.
Much? I don't know. No, now it's gone. Now it's dead. Okay. So where were we? Again, sorry.
So we were talking about the fact that there is less drugs coming out against GPCRs than there were before.
Yeah, but yeah,
Because
okay.
of our focus on, bias signaling, for example, and pathway 1 versus pathway 2. And in the past, people were not necessarily they didn't necessarily know the target. They went and saw an effect, did the animal studies, and then walked
Hmm.
did the studies.
Yeah,
And the idea was that we were focusing on getting a drug through the process. Yes, it would have side effects, but then it would have acceptable side effects.
yeah. But what you also said was the genetic variants, and this
Yeah,
is what, yes, yes, yes. That there are genetic variants, but I don't think that the knowledge of genetic variants will make us have more GPCR drugs. I think there are just so many new aspects to
yeah,
to influencing GPCRs with medicines that it's getting more complex and complicated.
yeah,
Yeah, but I still think that the two reasons why there are not more drugs is that because the receptors which have like major function on homeostasis are just exhausted,
hmm,
and we are getting too specific with our wishes, and
yeah,
making them the drug so special to be suitable only for very few people. And that means we have to identify these
yeah,
people, and that
yeah,
perhaps we don't manage. So, but this is, I mean, the whole field of medical treatments is getting more and more specific. So it's
yeah,
not only more difficult to find new GPCR drugs, it's more difficult to find new drugs at all because we have so many already,
yeah,
right?
yeah. Yeah, when I was talking about genetic variants, I was more thinking about having a drug and then knowing the genetic background of patients would determine whether the drug would be given or not, because we know that if it's how much it's going to act.
Yeah.
But if you're sick, you just want something to work. Even if it works just a little bit, you want to get better at
Yeah,
that.
yeah, yeah. But if you let's say you have high blood pressure, you're sick. Well, then there may be beta blockers, there may be angiotensin receptor blockers and
Yeah.
diuretics. So there are so many different things. And I
Yes.
think there already we have to consider that different patients have a different type of disease.
Yeah.
And on top of that, we have to then, depending on the target, consider the mutations. So it's just
Yeah.
getting more and more difficult, and we perhaps understand more and more why certain drugs only work in one but not the other patient, and
Yeah.
that we should be taken into account in the future when choosing the medicine, to give it to the right people.
Yeah.
Yeah, yeah.
Yeah.
But I mean, I would also like to see the pipeline of GPCRs be filled again, because we are educating people in our labs that have GPCR expertise. So it would be nice to know if they go to industry, they are needed
Yeah. Yeah.
because
No, absolutely.
GPCRs are in the pipeline. And when I came to Aventis at that time in the year 2000 or 1999, the pipeline was full of GPCRs and they have all disappeared.
Hmm.
They are all gone. They are completely fed up. They don't want anymore. One drug after the other failed. And then at
Yeah.
some point they are gone.
Yeah.
Yeah.
Yeah. I think we've come a long way when it comes to the tools and the understanding. And it's now an era where you need computational biology, you need machine learning in order to make sense of all of the data that we can acquire. I'm thinking here, doing cyclic AMP assays with radioactivity and then purifying your cyclic AMP, running it through columns and quantifying your radioactivity versus doing it in 1536 well plates and getting a ton of data. And sometimes you can get lost in that data.
Yeah.
All right, so let's talk a little bit. We've talked throughout our conversation about, giving some sort of advice to junior scientists who want to contribute to the field. We talked about, going away, going abroad, showing that you can adjust, you can grow, you can, form collaborations in a different country, in a different lab. Any other advice for junior scientists that you think might be useful?
That's difficult. I think they should follow their heart and what they are really interested in. I think because you have to be good and you have to compete. You will only be good if you do what you like. So make sure you do what you like. Don't go to a lab that publishes well but doesn't work on something you are really excited about. So that is an advice I have, just do what you like, because if you do what you like, you will be good at it.
I
That
think
is
so
one
too.
thing.
I think
Yeah.
so too, and you wouldn't feel like you're working. It's not like doing. You mentioned working in a pharmacy for 2 years and being bored out of your mind, and it was obviously not a good place for you to be in. I think being able to recognize that you actually don't like something is also very, very valuable.
But you know what is good? If you work on something you don't like, you are not in danger of working so much that you are actually destroying yourself because of the amount
Yeah.
of working hours you are putting in. So that
Yes.
at least is the positive thing. And then you have to choose, because if you like something, you are also automatically motivated to work more.
Yes. Yes.
Yeah. And
Yes.
that's where people should pay attention.
Yeah, I think so too. And I tend to get emotionally involved with the projects I work on because I like them so much. And I have to remind myself that, my value or my me as a person is not determined as how the project turns out. I have to think about drawing that line. But then when you like it so much, it's your baby, working on a project and then writing up the paper and, you know, responding to reviewers.
But this is normal. It's just normal. I don't think you can change it. And if you are different, you are then not like a real scientist. I think
Mm,
it's your project. It is just as it is,
yeah,
and then your baby grows and it's fine and it doesn't need diapers anymore. You put it away and you take the next baby. I think that's
yeah,
absolutely normal,
yeah,
because you have to be passionate about your project.
you do.
I
You
mean,
do.
so I don't think you can change this, no?
Yeah, I think that's the normal way of going about it. That's how, if you're passionate about something, it's part of you. You're always constantly thinking about it. Even when you're not in the lab working on it, there are parts of your brain that are focused on solving a problem that you're really passionate about.
Yeah, so to be passionate is something I would recommend junior scientists to be. And the other thing I recommend is that they should read papers and not only title or abstracts, and not believe what people are writing, but be critical,
Yeah,
look at the figures and try to draw their own conclusions. So, because often I see, and I'm doing the same, and I guess everybody does the same, because there is no time. You read titles and you think you know what is in the paper. You read the abstract, you think you know what is in the paper. Well, no, maybe what is in the abstract is the opposite of what you would think is in the paper. And this is why I would say to people, well, read your research careful and make up your own mind, and dare to have another opinion than others. And hopefully have your other opinion accepted by others with whom you discuss. So that I also think is something important, not just yeah, scan abstracts, scan titles, and accept what there is. Well,
yeah,
no, even if it's from big labs, it may not be what you would interpret into it, or perhaps others. So to be more circumspect about the conclusions you draw
yeah.
based on certain data. This is also something I would recommend people to do as long as they have time.
Agreed, agreed. I
Yeah.
think looking at the figures is very important to understand exactly. You read the title, you read the abstract, but take a look at the figures and to understand what was done. Do they have the right controls? Do you agree with their conclusions? then again, it's hard because at least when you work in the lab, you're drawn to doing experiments rather than reading. Although reading is very important, just as important as doing experiments. All right, last question, and I kind of flipped it a little bit. Top 3 aha moments that you had as a scientist that shaped your trajectory. Oh,
Saw this question before, and I thought, well, I don't actually know because I don't really know. I mean, aha moment sounds like a big discovery, and I'm not sure if any of the discoveries we have made are so big that an aha moment would
mhm,
follow. I think I was surprised about results, when I mean, I wanted to see arrestin signaling, so I have to get rid of the G proteins to finally see the arrestin signaling, and there is nothing. I only see G protein signaling. It's like an aha moment. I'm not sure if it's big enough to. So it's like there were findings that you obtain and you are surprised about the outcome. But I think an aha moment sounds like something Nobel Prize
yeah.
like, and I don't have that. So I have been surprised often, like the discovery that I think resins don't signal, but rather contribute to G protein signaling. That was one of the big discoveries I think that we made, and
Mhm.
of course it's of relevance for the ligand bias paradigm. But otherwise, I think so. You have many surprises, but oh, maybe I should say different. I think I don't have really aha moments because the research is not heavy and relevant enough. But what I discover through my scientific career is that the really cool projects are not the ones that I have thought about. These were accidents that you have to spot and then convert into projects. Because the projects you have and you think about are rather boring, and the aha moments are the surprises
Yeah,
that are actually the opposite of what you thought.
yeah,
And so, and this is then again an advice for young people: be open to interpreting your results different. So don't be biased and look what you are looking for, but be open that something is perhaps completely different, and then discover it and deal with it. Yeah,
yeah.
something.
I think that's very relevant. I think it's about, following where the data goes, following the science, and you never know where that takes you. You need to, think of a project, write up a project, start working on it, but then you'd be surprised where the data will take you, and you have to follow it. And no matter what the data, even if it's opposite, if the results are opposite to your own, thinking, you have to make sure that you obviously collected the right data, have the right controls. It is true what you're seeing, but also you have to follow it.
Yeah, because the aha moments you have when you don't expect what you get, and you have not, you
Exactly.
couldn't predict this before. Yeah, but I think they are still too small to be, presented
Yes.
here because it's just
Yeah.
always the surprises that happen all
Yeah.
the time.
Yeah.
These are the
Yeah.
small aha moments you have actually all the time, because you have a question, you do an experiment, and you have 10 new questions, and there are 10 little aha moments in there that no,
Yeah.
it's different, and it's just
Yeah.
open to accepting that everything may be different. Yeah.
No, agreed. Agreed. And on this note, Evie, thank you so much. We're going to stop recording in a second. Do not go anywhere. We're going to let people wonder what we talk about after we stop recording. But I really, really enjoyed the conversation. Thank you so much for being here today.
Thank you so much for having had the opportunity to be here today. And I look forward to some more minutes before I have to rush to my other appointment.
Yes, I will let you go very quickly. All right. Thank you so much. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our Dr. GPCR podcast guest and our team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. Please mark your calendars for our upcoming 2023 Dr. GPCR Symposia. Don't forget to visit us and check out the program. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem. Join us today. Also, please subscribe to the Dr. GPCR newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. With any questions or suggestions, please email us at hello@drgpcr.com. Until next time, stay safe.